Good afternoon or good morning, everyone. And now it's 2 o'clock. And I would like to welcome all of you to this stakeholder events. And EFSA is very glad that the public consultation is over. And so now today, and we are going to present you all the comments and also give an opportunity for you to discuss with our experts about how those comments and can and will be amended, addressed. So if you have any technical issues, and either do it by chat if you are really, really in trouble, and email Eric, his email address is on the slides. So how are we going to conduct this event? Next slide, please. Okay, so for those who have already attended those webinars in March, you are familiar with the rules. And for the newcomers, and this is just very brief, so there will be discussion sessions, and however, and during the presentation, so when the speaker is speaking, and your phone and your cameras will be switched off, handled by EFSA. And any questions arise during the presentation, please use the chat to send your comments. And if you can, ideally, you would put the name of the speaker, either first or family name, whatever feels easy for you. And then start with the name of the speaker and followed by your question. Please keep the questions short. If it's a long question, probably it's better to keep it and raise your hands during the QA session. And our chairperson will invite you to speak and raise your hand by that hand icon. And when the floor is given to you, please switch on your camera, unmute yourself, and give one word, introduce yourself, and introduce your question. Equally speaking, also while you speak, please try to keep your question really, really succinct. And also be reminded that today's session will be recorded and will be published on the EFSA website after this event. And so for the people during the registration did say that you do not want either your voice, your name, or any of your information get released due to the recording. And well, the only way to guarantee that is probably I would have to invite you to exit today's session. So with that said, and next slide, please. Let's get back to the science. So you can see that this guidance and following the mandate from European Commission, and we have come all the way and to where that stakeholder events is marked on the map. And there are still a few miles to go. And so by the end of this year, and you can expect this guidance will be finalized and make available back again to everyone. Next slide, please. So today's agenda, and right now I'm welcoming you. And later on, today's session will be chaired by our working group chair and Professor Andrew Chasen. And then we will follow the agenda points and start by European Commission, give us a few comments, and then we go straight into the topics. And I don't want to bore you with the title and the name. And the only thing I will remind you is that the first discussion session comes at 5 o'clock, lasts for 40 minutes. And so we will try to deal with all the comments related to the item 2, 3, and 4 in that discussion session. And then followed by a coffee break. And at 4 o'clock, please get back sharp. And then we will take the rest of the items, namely item 5, 6, 7, 8, 9. And then the second discussion session will take place starting at 5 o'clock. And I think with this said, and Andrew, the floor is yours. And have a nice session. Thank you.
Thank you very much. Well, E, I think you probably just done my job for me, so it's I don't have much to say. But I would like to add my welcome to that of E, to all of you who've made the time. I think the working group and the unit are very grateful that you're willing to give up 2 afternoons to join us. There's nothing worse than actually having spent nearly a year developing a document and then to have a complete lack of interest. So as I said, we're very grateful that you've joined us. What I'd like to do is just spend 5 minutes very briefly describing some of the objectives that we set ourselves at the beginning of this exercise, and has very much determined the shape of the document that you all seen, the new revised and provisional guidance document. If I can have the first slide, please. The very first thing we had to determine was genuinely who was our audience for this guidance document. That may seem very straightforward, but it's not quite as clear as it may first appear. And we settled very clearly that our primary audience, our main audience, was essentially you, people like regulatory affairs personnel in large companies, consultants, and anybody who actually has assumed the responsibility of the production of enzyme dossiers. And there is a consequence from having chosen that audience, because we have to assume that that group of people have a relatively high level of technical expertise when it comes to food enzymes. And that very much determines the way in which we then write a guidance document. And secondly, the second objective and principle we set ourselves, if I could have the next slide, would be that our guidance document should be a stand-alone document. So when it comes into force, it will replace all other previous guidance documents and statements that have been issued at various times by EFSA and its panel. And it should be possible, this was our intention, it should be possible to construct the technical elements of a dossier from our guidance document alone. Now, of course, you will find references in this document, particularly to other EFSA documents. I think you're probably all very well aware that EFSA is very active in developing risk assessment methodologies. It also seeks, as far as possible, to harmonize its activities across its various panels and its working groups. So you will find reference to a number of EFSA documents, and these are intended both as background documents should you wish to explore further any particular area, but it will also in many cases give you the reason why we're asking for the specific data we are. So for instance, there is a cross-EFSA genotoxicity working group, and as far as possible, it's expected that the panels and the working groups would follow the recommendations of the genotoxicity working group, which is what we do in this document. It's not necessary to go to the specific document. We have all of the information necessary in the guidance, but the added information is there if you need it. And the next, please. The other thing we recognize is that we should, wherever possible, as I said, indicate why we ask for specific data. This enables you to perhaps interpret our intention and provide data in a more effective way than we have specifically asked for. But we also recognize that no guidance document can ever be totally exhaustive. We've been surprised already by some of the applications we've received over the year. So we have introduced some flexibility into the document in the sense that if you believe that a data request included in the guidance is not applicable to your particular case, then by all means provide us with a scientific justification why it should be omitted. This is fine, it will be considered. And similarly, there may be occasions because of an adverse response in a study that we need to follow up and ask for data which may not be necessarily specified in the document. And finally, we decided quite early on that the structure of the guidance document should reflect the structure of the food enzyme opinions. We have those of you who are familiar with our enzyme opinions know that we have a constant structure based on essentially on 5 elements. That's the source of the food enzyme, its manufacture, characteristics of the food enzyme, the toxicological data, including allogenicity, and dietary exposure estimation, finishing, of course, with a conclusion. And by and large, we found that this structure works well for virtually all of the applications we've received to date. However, we did take pity on those of you who want to produce plant or animal enzymes, in that we've hived off these sections on the basis that probably you did not want to wade through 20 pages of instructions for those with a microbial enzyme before coming to the mention of the word plant. So we hope that this is more user-friendly. It has a downside that it meant there's a little more replication, duplication in the guidance than is strictly necessary, but we think this is probably worthwhile in terms of clarity. But I'm sure you'll let us know if that's not the case. And then finally, we have part B. And this is one of those sections where we actually really do need your feedback, because of course this whole section has been written without ever having seen a relevant application. Now this will be the topic for discussion for tomorrow afternoon, but as I said, this is the one area where this is not based on experience, and we're guessing on the data requirements that be required, so we would very much like to hear your views. So those are the principles, basically, or the objectives that we had at the beginning of this exercise. If I could have the next slide, please. It really is now down to you, I think, to give us the feedback that actually will allow us will inform us whether we've actually succeeded, or the extent to which we've succeeded in our objectives. So that was a very brief overview over our approach to the development of this guidance document. I think now is the time we should actually move on with the discussion. And we're very fortunate in having Catherine Evrin here from the Commission, who is responsible for the structure and the construction of the future community list of food enzymes. So without more ado, I would like to pass the floor over to Catherine, if she's there.
Good afternoon. Good afternoon, everyone. I think you can see me and hear me. Thank you very much, EFSA, for organizing this meeting. I will try to share my presentation. Do you see my presentation?
Not yet in the presentation
Not
mode.
yet. Okay. I think I'm
no,
not.
We see it, but not in the presentation mode.
Okay. Okay.
Yeah.
I think it's okay now.
Yeah.
It's fine now.
You. Thank you very much. So I'm in the team in charge of food improvement agent at DG Santé, and in particular, I'm dealing with the issue on the food enzyme. So today, I would like just to remind you that food enzyme are covered by general horizontal legislation as other food. General food law and food hygiene, and also official control in the case of the control authorities. And there are also a specific legislation for food improvement agent, which covers additives, enzyme, and flavorings. And in particular, the common authorization procedure regulation 1331 of 2008, and its implementing act, where the risk management and risk assessment data are detailed. On food enzyme, the legal basis is Regulation 1332 of 2008, and it covers food enzyme used for a technological purpose in food and also in novel food. And it doesn't cover food enzymes which are used for a digestive or nutritional purpose. They are excluded. Food enzymes which are used in food supplement, for example, are not covered by this regulation. So this is for the scope, and it includes a requirement to establish the register, a union list, and also a provision for labeling. Risk management matters are discussed by the Commission and the Member States at working party with the experts from different countries. And in general, we have 2 meetings per year. As regards the topics that we discuss at this meeting, you can see on this slide the agenda of a standard meeting of the working group. And I will use this agenda to go to the different topics and give you some updates of our works. As regards the state of play of food enzyme dossier submission, up to 2015, there was more than 300 food enzyme application that was considered valid, and they are now included in the register, which is on the website of DG Santé. And also, I would like to add that this is not a union list. So the register, all the food enzyme and on the market are still under the national law. Some competent authority has some provision for food enzyme. So the food enzyme could be on the market according to national provision. Since 2015, we have received 44 new applications, and recently 28, so at the beginning of 2021, before the entry into force of the transparency regulation. And thanks to AMPEP, we have been informed that 37 dossiers are expected to be submitted in 2021 and 50 in 2022. So for the application in the register, we have negotiated a deadline for the evaluation with EFSA, and there is and regularly EFSA informed the Commission on the progress of the work plan. And for a new application, in that case, 9 months period apply to this new application. And if you follow the adoption of the opinion, you can already see that some of the new application, the opinion have already been published. With the entry into force of the transparency regulation, which it will be detailed more tomorrow, a new e-submission system is in place since the end of March this year. As this regard, I would like to say that some of the application are not submitted to the new transparency regulation. For example, the data package for the joint dossier, they are not considered as new applications since mandate have been submitted years before. And also, if applicants want to submit spontaneously new data to improve a dossier already submitted, we are not in the case of new application, so there is no application of this transparency regulation. I would like also to add that if there is an amendment of the source of the food enzyme, in that case, it is a new application. It is not considered as an improvement of the initial dossier. And we have some cases where the applicants are no more interested to put on the market the enzyme which has been introduced in the register because they have developed a new enzyme. So in that case, they withdraw the application and submit a new application. So I just would like to say that if you are in this case, withdraw your application as soon as possible because it saves the time for EFSA in particular to assess the other application. There will be also a mandate soon to EFSA to reopen the exposure part of certain EFSA opinion before the application of this of the new exposure system. So we need to ask EFSA to update the first adopted opinion to have the application of the same tool for each. As regards the establishment of the union list, it will be done in a single step once EFSA will have issued an opinion for all the food enzyme in the register. So there is no positive opinion yet. And there is no date indicated in the legislation. So all EFSA regularly updates the plan and to deliver all this opinion as soon as possible. In the meantime, there are opinions for new application, and this opinion will also be considered in the work to draft the union list, and they may be adopted in the same time or very soon once all the food enzyme in the register will be assessed. As regard the union list, as you can see, the regulation provide some information as regard the content of the list. And for this to draw this union list, the member states oh yes, we work with the member states on two other topics. EFSA gives its opinion on the safety concerns, but we have also to deal with the technological need and also that the use of the food enzyme doesn't mislead the consumer. And this is done with the member states on the basis of the dossier. So how appraise the technological need and the misleading to the consumer? So it was decided with the expert of the working group to adopt a general approach and to assess these topics processes by processes, and we have started to open the processes of fruit and vegetable processes and also wine processes. As regard food and vegetable processes, we focus first on the fruit juice production. The reason is that for this two kind of commodity, there are some regulation which already allows a list specific food enzyme, and so we have started to look at the correspondence with the application and the provision already set in the regulation for this food, and the necessity to update in some cases the regulation in parallel. We also, in parallel, start drafting the EU list based on published opinion, and we also try to go with a group of food enzyme, and this is the case for lipase, for which EFSA has already issued a number of opinions. As regard the working document describing the food process, this working document is important to harmonize the description of the different food processes in which food enzymes are used and subject to authorization. So it is a useful document for EFSA. For the assessment and the exposure, and also for the drafting of the union list. So this working document has not been published, and it is under review. There was an important update at the end of 2020 to take into account the progress made by EFSA in the assessment of several processes. So the idea is to keep align the document published by EFSA for the exposure and this working document, which can be used by the applicant for the new application. I would like to thank the stakeholder for their feedback that will be shared with the member states during the next working group. There was no major modification, but improvement of the description, and we will take into account the proposal made by the stakeholder, and continue to improve the document also based on the assessment of the dossier with the member states. So I have finished for this information. So thank you, and I think I stay online if you have further questions. Thank you.
Thank you very much, Catherine. Yes, there will be an opportunity, I hope, to discuss with you during the question and answer session in about half an hour's time. And I'm sure that you have raised a number of issues of interest to the various stakeholders. But now at the moment we move on with the EFSA guidance. And as E has already indicated, we have a number of presentations by members of the working group. In each case, they will attempt to answer some of the questions that we have already received. And the first presenter from the working group is Jose Barra-Bavera, Professor Jose Barra-Bavera, and I leave the floor to him. Thank you, Jose.
Okay, Andrew, thank you very much for what. Sorry? Sorry, Andrew. I thought it was Pierre Sandra before me.
So he went through quite all the.
It's you, Jose.
He stopped after. Yeah.
Okay, thank you. Well, thank you very much. I'm going to do the short presentation related to the production and the characterization of the food enzyme of the guidance. And just want to specify that I will be focused on the use of microorganisms as source of the enzyme, and the main differences will be presented afterwards by my colleague Holger Sohn. Sorry?
If you can stay here, of course.
I think somebody is unmuted. So
listen and wait for the question.
sorry, Catherine, could you mute, please?
Yes, I have mute, Catherine.
Yeah,
it's fine.
Catherine. Yeah.
Oh no, now she's unmuted. Okay. Well, as I told you, the main differences between the microorganisms as source of food enzyme and plants and animals will be explained by my colleague Holger Sohn. As regards the production of the food enzyme, we have the presentation is divided in the section related to fermentation and downstream processing, and I will devote some attention to explain specific aspects of the food enzyme preparation. Well, fermentation is important in the safety assessment because as a consequence of this step, the microorganisms or parts of them will end up in the food enzyme. In addition to that, the metabolites produced during the fermentation will be also present in the food enzyme. And finally, the products used during fermentation can end up in the food enzyme, and in some cases, some concern can exist, such is the case of the presence of antimicrobial resistant genes, mycotoxins produced by microorganisms, or for instance, the potential presence of antibiotics that can be used during the fermentation step. In addition to that, the food enzyme will contain a lot of unknown compounds coming from the microorganisms. And that's why we use the TOS concept, the total organic solids, which is the totality of the food enzyme with the exception of water and ash. And in some cases, we could accept the use of food enzyme preparations in the dossiers. And in this case, the TOS will be calculated, will be the whole food enzyme preparation with the exception of water, ash, and the total added organic excipients. If you go to the following slide, please, yes. In case of downstream processing, it is related to the food safety of the food enzyme, not only because of being a potential source of substance of concern, but also because it modifies the properties of the fermentation products, in general, making them safer because they are used for killing the fermentation microorganism and for purification purposes. Please go to the following slide. So, in general, we need the full details of the production process because we need to know what is happening during the process. So to have an idea of any concern in the food enzyme can be derived because of the process. And that's why we need to know the flow chart. And in case of food enzymes produced in different places, we need to know if there are differences in the production process that could affect to the purity or the composition of the food enzyme that is under assessment. We also need the chemical identity of the substances used during the fermentation of the product, and the analytical data, which may be required in case of safety concerns because of potential carryover into the food enzyme. And finally, we need a statement that the food enzyme production meets the food safety management system principles and is in accordance with the food hygiene regulation. In case of food enzymes produced outside the European Union, we need confirmation of the equivalence of the production process principles with the European Union regulation. Please go to the following slide. More specifically, in case of the fermentation, we need to know the type of fermentation system used, and we need the list of the raw materials, reagents, and processing aids that are used or might be used during the fermentation process. It's important to note that an indicative list will not be accepted. In fact, this is one of the reasons for going back to the applicant asking for the list of the actual materials that are used during the fermentation, because they give us a clue or information of potential carryover in the final food enzyme. Please go to the following slide. As regards the downstream processing, we need a detailed description of the method used to kill, disrupt, or remove microbial biomass in case of existing this step, and to concentrate and purify the enzyme liquid. And as in the case of fermentation, we need a list of all the substances used including the processing aids. And just to remark that an indicative list will not be accepted. Please go to the following slide. As I mentioned before, the food enzyme is in the majority of the cases the product that is being assessed and which information is in the dossier. But in some cases, the food enzyme preparation may be accepted. And this can occur in case of food enzyme that is inherently unstable, and the only data on stabilized preparation can be generated. Or in some cases, when there is potential concern of the presence of carryover of hard source materials into food as a consequence of the preparation of the food enzyme preparation. Please, next slide. In the first case, we need the quantitative data of all the added excipients because it's needed to determine the dose content of the food enzyme preparation. And in case of potential carryover, in most of the cases, it's because of the immobilization or encapsulation of the food enzyme because of the supporting material or cross-linking agents that can end up in the food enzyme or in the processed food. So that's why we need to know if there is presence of this carryover in the food enzyme or analytical data of the potential presence of the carryover or the cross-linking agents in the final processed food. Please go to the following. Now we have seen up to now the production of the food enzyme and the information needed, and now we begin with the characterization of the food enzyme. It has 4 sections that are the ones that we have in the slide. The first section is the properties of the food enzyme. Please, can you go to the following slide? As regards the food enzyme, we need to know the amino acid sequence of the actual enzyme. The reference to published sequence of enzymes with same catalytic activity are not acceptable. From the amino acid sequence, we need the molecular mass calculated from this data. And we also need the protein pattern of at least 3 batches of the food enzyme and all the additional batches that are used for toxicological purposes. All this information is used, among other things, to determine or to check if the food enzyme batches used for toxicological studies are suitable because are comparable with the food enzyme used for commercial purposes. Please go to the following slide. We also need to know data of the enzyme activity. First of all, we need a description of the standard operation procedure used for measuring the activity. Here it is important to state that we need to have the same type of units for enzyme activity to compare the data from different batches of enzyme. We also need the temperature and pH range with activity of the enzyme, the optimum values for pH and temperature, and we need the thermal stability of the food enzyme within a range reflecting the technological role of the food enzyme. And just to state that we don't require to provide data on long-term stability of the food enzyme related to the storage. Please go to the following slide. As regards the chemical parameters, we need to get those parameters from at least 3 batches of the food enzyme representative of those intended for commercialization. From those batches, we need the enzyme activity or activities, as I told you, with the same type of units, the units per gram of batch. We need to know the total protein, ash, and water, and from those data, the dose calculated and the enzyme activity per milligram dose of the food enzyme. In addition to that, we need the same data set for all additional batches of food enzyme used for toxicological or other studies. All this information is used to determine if the food enzyme batches used for toxicological or other studies can be considered representative of the food enzyme used for commercialization. And just to remember that in all cases, we need the methods of analysis and the certificate of analysis. The lack of existence of certificate of analysis is another of the reasons why we have to return to the applicants asking for information. Please go to the following one. And now we go to the purity section. In this section, we need to analyticate data on the concentration of medium ingredients added for purposes other than nutrition or pH control, the concentration of lead in the food enzyme, the concentration of compounds of known toxicity, such as potential presence of mycotoxins. In fact, these mycotoxins can be, the potential presence can be obtained or checked through literature searches of the whole genome sequence analysis of the production strain or from literature. We need to know about the microbial purity of the food enzyme, and specifically the presence or potential presence of E. coli and Enterobacteriaceae. And finally, the antibacterial activity of the food enzyme that should be determined according to JECFA. And again, it's important to remind you that we need the methods of analysis, certificate of analysis, and the limit of detection and limit of quantification of the different analyses. Please go to the following slide. The last of the section of the properties of the food enzyme is that related to the presence of viable cells and DNA of the production strain. And in particular, I'm going to give some information as regards viable cells of the production strain, because all the requirements related with the determination of DNA from the production strain will be explained by Professor Pier-Sandro Coconcelli. Please go to the following one. This is my last slide. Viable cells of the production strain must be determined or measured in all the food enzymes except for those obtained from not genetically modified QPS production strains. And the technique used to remove the microbial cells must be given, in particular during the downstream processing step. And the determination of the viable cells must be culture-based, enabled to recover of stressed cells. It has to consider the specificity against contaminating microbiota. If the strain is able to form a spore, their possible presence should be analyzed. It's important to state that we need a minimum of 1 gram or milliliter for doing the determination of microbial presence, and at least 9 samples obtained from a minimum of 3 independent batches are needed. And finally, a positive control with samples of the food enzyme must be added to the analysis. And then go to the following one, and with this last slide, I finish my presentation. So thank you very much for your attention.
Thank you very much, Jose. Okay, I think we need to move on with the second presentation in this section, and then we will break for a Q&A section when you will have the opportunity to follow up the comments that Jose and Holger and Catherine have made. So the next speaker in this section is Professor Holger Zorn, and he'll be dealing with one of the interesting areas of the dossier, and one which has been expanded very substantially compared to the original guidance document, and that's the question of enzymes of plant and animal origin. Holger.
Thank you, Andy. Do you see my presentation?
Yes, I do. Thanks.
Yes, dear colleagues, as Professor Chesson already indicated, we decided to keep separate sections for enzymes that are derived from animals and plants, just for the simple reason that some of these enzymes have been used for decades or even longer, like chymosin, and most of the enzymes are in fact derived from food as it is normally consumed. And this is especially true for all of the animal and plant derived enzymes we have evaluated so far. This is the current list. We had already 2 enzymes derived from the class of redox enzyme, oxidoreductases, one ascorbate oxidase from cucurbit, one peroxidase from soy. Then we had a couple of beta amylases, for example, from barley, from soy, or also from wheat. We have evaluated one lysozyme from hen's egg white. And only recently, we have evaluated a couple of pancreatic enzymes, including chymotrypsin, elastase, and trypsin, that are derived from pig's pancreas. Another traditional enzyme that is used widely is chymosin from the abomasum of cattle, of sheep and goats, and also those have been evaluated only recently. The working group has received a couple of really thoughtful comments by the stakeholders, and we are really grateful for your careful reading and the many suggestions you have made. Some of these suggestions address an especially interesting point, namely food enzymes that may be derived from genetically modified plant organisms, and the stakeholders, as in the public consultation comment was indicated, recognized that they are really handled in a different way from that what we do with the GM microorganisms. And the same is essentially also true that GM animals have not been considered in our current guidance document. The reason behind that is considered of two answers. First of all, we of course wanted to draft a guidance that is fit for purpose. And so far, we just have not received any applications for GM plant or GM animal enzymes. So that is one reason. And the second one, maybe even more important, even more stringent, currently those enzymes that are derived from GM plants or animals that have not received authorization according regulation 1829 are currently just not within the remit of EFSA. So those cases would first need a discussion with the Commission. That is the reason why the working group now proposes to change the wording that was originally presented in the guidance to just one sentence, which reads the same for both GM animal and plant enzymes. It should just be indicated whether the source is from a GM animal or a GM plant, respectively. If we get such opinions in, of course, this has to be discussed with the Commission. And from a scientific point of view, it seems somehow logic to handle these enzymes analogously to those of enzymes derived from GM microorganisms. But again, this topic needs clarification with the Commission. The guidance is thus rather short for these two parts. As indicated previously, we need the source of food enzymes from plant and animals. We have the production section, including extraction, DSP, and if applicable, also the immobilization. Then we have the whole set of characterization, including the properties, chemical parameters, and especially, of course, the purity is relevant here. One comment we have received is a rather old topic that had been covered by previous guidances of the working group, namely site activities. That was a long and an old discussion in the working group, and after having discussed this topic now for essentially a bit more than 10 years, some of the working group members are working for more than 10 years in this field, and thus far no topic came on our table where this topic was relevant for safety evaluation. So the working group for now decided that we, especially also considering that most of these enzymes are in fact derived from food material, there is no additional value in reporting site activities. Then there were a couple of comments that are scientifically highly interesting. Namely, one stakeholder suggested that the temperature of complete inactivation, as indicated by activities assays, should also be reported with the temperature of complete denaturation of the enzyme, which means the unfolding. Of course, it is scientifically correct that both temperatures may not be exactly the same. And apart from that, one stakeholder also asked for the activity analysis in the final food as processed under industrial conditions. Of course, it's a quite well-known fact that an enzyme may be much more stable in the real substrate where it is used industrially as compared to a buffer system. But nevertheless, our entire evaluation addresses enzymes in its active state, in their active state. So from a safety evaluation point of view, it does definitely not make a difference if the enzyme is inactivated, denatured, or in its active state. This is so not of additional value for safety evaluation, and there is no need to provide these data. The same is true for the inactivation under industrial conditions. One reason is that, of course, that's much more difficult to measure, also considering the fact that you may have endogenous alpha amylases plus added alpha amylases. But again, from the safety point of view, it does not make a difference. This point may somehow become relevant later on if it comes to labeling decisions. But for these decisions, I think the data, as we ask for them now, are fine anyway. As already indicated by my colleague Jose, we don't require data on long-term stability of the enzyme. That is not of help for safety evaluation. The story is somehow different for potential contaminants. One stakeholder asked for quantitative values for lead, cadmium, mercury, and arsenic, and after having discussions and also going back to different types of literature, the working group proposes now to adjust our text and to include threshold values for the four named metals of 0.5, 0.5, and 3 milligram per kilogram of food enzyme, as it is currently already the case, for example, in the French regulation. This is based on the fact that we have observed contaminations in plant-derived enzymes, and these levels are considered safe. So I think that is of help for the applicants to have clear guidance here. Another comment on the purity section addresses the potential presence of pesticide residues. As it is worded currently in the guidance, the working group, of course, would like to see potential residues of pesticides if pesticides have been used. There was one proposal to restrict this analysis only to those pesticides that have not been approved for application in the European Union. But of course, this is not the meaning of our guidance and not the meaning of that what we intended. Of course, the working group would like to see data, analytical data on the potential presence of residues whenever they are used, independent from an approval in the European Union. So for safety evaluation reasons, we always ask for potential residues of pesticides whenever there was a potential use of them. Now we switch to the section of animal-derived enzymes. The first proposal that was made by one of the stakeholders was to extend the search for absence of risk on infectivity, namely to include the potential risk of TSEs and of parasites. We had quite intense discussions in the working group on these suggestions, and after the consultation also with the colleagues from veterinary medicine, we decided not to include this topic here, the first one, because the absence of parasites is typically guaranteed by veterinary inspection, but also by the different steps in the production process, like, for example, freezing or filtration processes. Also, the second topic was not regarded as currently being relevant for the guidance, as typically no risk material like brain tissue or bone tissue is used for enzymes derived from animal origin. So of course, this would not mean that we do not keep the freedom to ask for that. Whenever an applicant would, for example, propose the extraction of cattle bone for preparation of an enzyme, of course, this topic would come back, and in this case, we would certainly ask for the presence and or absence of prions, but not for the current guidance, because it's currently out of the scope. A very thoughtful comment was received on the section of potential microbial contaminants present in an animal derived enzyme. The suggestion was to include an additional or inactivate in the guidance. And that, of course, does really make sense. The working group was happy to receive this comment, and we accordingly have adopted our wording. And now the sentence reads, the specific methods used to remove the animal tissue after extraction, to concentrate the enzyme liquor, and to remove or inactivate microbial contaminants. So this is really helpful, and of course well reflects that what is really done in the production process of animal-derived enzymes. There was another suggestion which somehow makes sense, namely to align the wording that is now present in the plant-derived enzymes and also in the microbial enzymes, namely the suggestion to include the paragraph, the protein pattern characteristic of 3 batches should be provided by, for example, as this page or size exclusion chromatography, of course also including additional standards. However, having seen now quite many of the applications for animal-derived enzymes, for example, of pancreatic enzymes, this just does not make sense, because the chromatograms, based on the nature of these enzymes, are just overcrowded. So you may see an SDS page, but you just don't see any bands. It's more or less brown or black gel, depending on the staining that has been used. So this is not of additional value. And also, again, considering the nature of the food enzyme as being derived from food, this was not considered to be necessary. So in this case, for the animal-derived enzymes, we do not ask for SDS page analysis. And again, also on this topic, normally we ask for the degree of glycosylation, which has been changed also according to a suggestion of the public consultation. We typically ask for degree of glycosylation, but not for animal-derived enzymes. With this, I hope to have addressed the most important topics for animal and plant-derived enzymes, and thank you very much for your attention.
Thank you very much, Holger. That was very thorough. Well, now we come to the point where you have the opportunity to ask any questions that you may want to ask. Could I suggest that we start with any questions you might want to address to Catherine and the Commission? I'm surprised if there are none at all.
Andrew, if I may, and there is a question in the chat box from AMFEP and Chelin. May I invite you to introduce your question verbally?
Yes, sure. Good afternoon, everybody. So I'm Céline Benny, the Secretary General of AMFEP, the Enzyme Association. So thank you very much, Catherine, for this for your presentation. And I would have 3 questions actually. The first one would be: so do you expect that the new guidance document will have an impact on the calendar of risk assessment? I also wanted to know, because you talk about EFSA calendar, what is currently the expectation for finalization of the assessment of the 300 dossier received, including the data package from the joint dossier, and consequently, if there is any tentative date for the publication of the union list. Thank you.
Well, there are a few easy questions for you, Catherine. Catherine, I think you're muted.
You're still muted. Yeah.
No, I think that she's not muted, but it's very, very low, the sound.
Well, she's staring into space, so I don't know.
Is it better now?
It's a little bit better, yes.
Okay. Yes, as regards the first question, the impact of the guidance, new guidance document, I think it will not have a negative impact on the calendar of the risk assessment. This new guidance gives some clarification of the expectation from the experts, clarification on the data, and it is an of help for the applicant to maybe improve the quality of the data that they have already submitted. And by consequence, it could reduce the time of evaluation of the dossiers that are in the pipeline. So I don't think this is a negative impact. As regards the finalization of the evaluation of the dossier in the register, it's very challenging for EFSA. We have this COVID situation, which has slowed down the progress last year, and also the splitting of a joint dossier, which increased the number of applications. As far as we understand, EFSA is always trying to improve the way of dealing with this dossier, but the tentative date of 2023 could not be a guarantee at this stage. But I have not other expected date to give to you. It's and for the publication of the union list, as I said, we try to work in parallel with EFSA in order to follow the work, the progress, and be ready as soon as possible when the last opinion in the register is published. Yes, I stop here. I think there is another question, but it's for another topic.
Thank you.
Celine, was that okay?
Yes, thank you very much.
Would you want to make a follow-up?
No, it's okay. So thank you. It's a bit of clarification.
So is there another question to Catherine?
May I invite Fred van der Germ?
Yes, hello. Thanks for your presentation, Catherine. You mentioned that some EFSA opinions are to reopen. Could you share some background on that and whether there might be more in the future?
I was mentioning this as regard the adoption of a new methodology for the exposure assessment. There was a statement published in 2016. The budget method has been replaced by a more realistic exposure scenario. So we decided to update I think there are 5 opinions that was published before 2016. And the purpose is to update the exposure part of this opinion in order to have all the food enzyme assessed under the same protocol.
Okay, so it's just about the availability of the PHI model and no other reasons there.
Yes.
Okay, thank you.
Yes, we are actually hoping that those requests will not come through until we have all of the PHI models in existence, because then it should be a relatively straightforward activity. Okay, so no other questions to Catherine?
There is a hand raised, Andrew.
Okay.
From Mariela Kierman.
I can't.
Yes.
Got it. Yes.
Okay, thank you very much, Marielle Kalm from Amphep DSM. Well, thank you for the very interesting presentation, and it gives us some more ideas on the progress and the process. You indicated that the positive list will only be published when all the 300 dossiers have been evaluated by EFSA, and of course then there will be more time from the Commission and the Member States to work on that. It was also indicated that new dossiers, you have a kind of agreement, how I understood at least with EFSA, that they have the legal 9 months to assess those new submitted enzymes after 2015. Does that mean that as long as those 300 enzymes have not been evaluated, that as long as we submit dossiers 9 months before the end of this period, that they will be taken into account and that they may appear on the first positive list?
This is, yes, the idea is to deal in parallel with this new application and the enzyme in the register, and to take on board as many as possible food enzyme which are valid dossier and for which it is possible to make progress to publish them in 1 or 2 steps, but very close, in order to have a smooth application, a smooth entry into force of this union list. And so I cannot guarantee if it is the period of 9 months before the last the assessment of the last enzyme, but the idea is to do this way, and until the last food enzyme from the register is not assessed, to accept the other in the workload.
Okay, yeah, thanks a lot. I fully understand that you cannot guarantee anything. I think the only thing that we would like to ask is if we get at least an idea of the timing in advance, so that we can put our own working scheme in line with that.
Yes, as I said, there are many. There have been many disturbances in the process, but we don't have a clear date for the end of the process. We can. EFSA is prudent also in the planning, work plan. It depends also on what the applicant, the speed at which they will provide additional information. So it's not really, it's not possible to give you a real date. There are many uncertainties.
Yeah, I fully understand, and I think it's very good that we have today the discussion, and as long as we keep talking with each other, I think we will manage together to come to a good end of this whole exercise.
Indeed. Thank you, Mariella. Okay, if there are no other questions to Catherine, perhaps we could turn to the elements of the guidance, which deals with the characterization and manufacturing processes, perhaps of microbes, plants and enzymes. It's plants and animal enzymes.
Yes, Andrew, and there is a comment, actually 3 comments, from Elisa Lawson, from Christian Hansen, and can you please repeat your question? Thank you.
Yes, sorry. So hello, Elise Lawson, Christian Hansen. Yeah, so this is both for microbial and for rennet. When you're talking about the production on different production sites, and the guidance asks for a very, just for all the differences between those production processes. So what I'm fishing for is what kind of detail are you looking for? Is it acceptable to describe only the differences which are expected to have a significant effect on the purity or composition? Or really every small detail, which could be a lot. The second question is really for microbial enzymes. It's asked in the guidance which methods are used to kill, disrupt, and remove the production strain. I think we were several producers wondering why it was asked, the methods were asked. Why we should provide details on how the strains are killed or disrupted. Is it not enough? Is it not the most important thing how they are removed? And finally, about providing specific raw materials, that is something that can change. Sometimes in companies we need to replace or remove a raw material in our production, or even introduce a completely new raw material. So we're just wondering what the expectation is there. Whether every time we change, I don't know, should an update be sent, or should we make sort of an evaluation within the company of whether or not the change is significant and has an impact on the final enzyme? Sorry, those are my questions.
No, they're 3 very good questions. Holger, do you want to try and take that on first, and then Jose?
Maybe the first one, yes. That was the difference of the so-called primary production site that was indicated in the original guidance document. We have taken up this comment and have slightly changed the wording. What is really meant here that you should describe one production plant in detail, and all differences that may have a significant impact on the safety should be indicated in detail. So if you use, for example, a different DSP for a microbial enzyme, a different filtration system, that of course may impact the presence or absence of recombinant DNA or the production organism in the final food enzyme. So this would definitely make a difference. A minor change of 1 or 2 degrees centigrade in the fermentation for those enzymes would certainly not be meant with those differences.
Is it fine with the first question?
Yeah, that's okay. Thank you.
Okay, if you want, I can answer the following one. As regards the need to know the method used to kill, disrupt, and remove the production strain. Well, first of all, just to mention that in not all the cases the production strain are removed. Sometimes they are killed or disrupted and they remain in the food enzyme, the remains of the microorganisms. And on the other hand, sometimes the companies use killing agents for killing the microorganism. We need to know this information just to know if any potential carryover of the killing agents can end up in the food enzyme. So yes, the answer is yes. We need all the information of all those methods.
Maybe I can just add one comment on the killing agents. Of course, we have seen a couple of rather normal killing agents, but for example, some companies also use formaldehyde for killing off their microorganisms in a whole cell system, for example, and that of course is a topic that may raise safety concerns and therefore has to be addressed during safety evaluation. It does not necessarily mean a risk, but of course the topic has to be addressed.
So you're specifically concerned about the raw materials that could be used as killing agents or disruption agents?
Yes, and if the use process has enough consistency to assure that the microorganisms are dead, just to have a clue about what is expected in the food enzyme.
I think that's why we ask for all raw materials used in production, whether in the medium or extraction systems and in the downstream processing, because it allows us to make a judgment as to whether there is a concern there or not. But I think generally we just want a qualitative list. We're not overly concerned about carbon sources, nitrogen sources, pH control, these sorts of elements that may be present. But we are much more concerned about the other elements which may be carried through to the final food enzyme. We just want to be in a position to know what's going into the system so we can judge what the likely outcome is. But I think you raised an interesting question as to, I think this is a very general question you've raised, because I think we all recognize that the enzyme industry doesn't stand still, and that you will probably constantly be refining your production systems, including the way in which you grow a microorganism or extract an enzyme. And so you raise an interesting question as to, as you change over time, at what point does this become a different process? Holger, do you want to think about that one, or Jose?
Well, in my opinion, the answer is when a significant change occurs in the composition that may affect the result of the safety assessment. And as regards to raw materials, something can affect the evaluation of the allogenicity, because some of the raw materials have potential allogenicity, and when you change the raw material, sometimes it has to be indicated in the label. So I think many different circumstances can happen.
You may have seen in the draft guidance that we tried to include some freedom for the applicants by mentioning that for those compounds that are typically used just to supply the carbon or nitrogen need, or also the vitamins and minerals, we generally do not ask for any quantitative data. So changes in this area would certainly not be regarded as safety relevant. But of course, as Jose already indicated, if you use completely new raw materials, then this of course would make an update necessary.
And I think the interesting thing is it's now in PAX on part B of the guidance, because there is a requirement on all producers or users of enzymes to notify the Commission when there is a significant change in the process for an authorized, for an enzyme which has already received authorization. And the question is, and I think I suspect there will have to be discussions with the Commission as to what extent the changes are going to be before there is you need to trigger a legal requirement to notify the Commission. And then we have the additional point that the Commission is not automatically required to consult EFSA on this system, but only when they consider there is likely to be a health impact. I think we're dealing with an area which is as yet pretty much unexplored.
Yeah.
Do we have any other questions for this first part of the presentation? I'm scrolling through the hands and see none raised. E, do we have any other comments?
Yes, there is another one. Is Mariella from DSM, please.
Mariella.
Yeah, sorry, we divided.
Welcome back.
We divided the items, and this was my item. So, yeah, well, obviously we already indicated in our comments something on the micro specs. So I think you you will have expected this remark. We are wondering why the general microbial specifications are proposed to be changed. We had the specifications on E. coli, Salmonella, and oh shit, the third one. Well, now you are coliforms. Thank you. Yeah. Now you are proposing to go to a spec of 10 CFU per gram for Enterobacteriaceae.
Yeah.
And yeah, first of all, we are really wondering what is the background of this. And second, I think, well, I would after that elaborate a little bit what the consequences would be to change specifications. Now we already have submitted and are submitting dossiers based on the current specifications. So if we need to have change that, it will have a lot of implications.
The first one I can answer very quickly. I mean, clearly the new guidance has yet to be formally adopted by EFSA, and there will certainly be a grace period before it formally comes into effect. So I think in terms of the dossiers which have been submitted, or are probably those that are currently in preparation and ready for submission, this is not likely to have any impact whatsoever, because we would accept the 30 coliform CFU. We're talking about the future, and I'm hoping that Piersandro Cocconcelli is online, because
yes, I am.
He was responsible for this change, so you can defend your position, Piersandro.
The concept of introducing, and I hope also that Live, she's here as well. The basic concept behind is to harmonize the enzyme regulation with other food. So, as you probably are aware, since 2006. Or even 2005, maybe, with the regulation on microbiological criteria, European Union has shifted from coliform to Enterobacteriaceae, which is a more reliable value in terms of hygiene of the products. And so that's the reason why we have chosen to consider Enterobacteriaceae.
Well, could you then refer to which legislation that is? Because we are not aware of this.
It's a legislation 2073 of 2005 and the following modifications, in which are defined the microbiological criteria, both food safety criteria and the process hygiene criteria for food.
Well, as far as I am concerned, this is only for very specific foods and not for food in general. So in that sense, I mean, when we talk about harmonization, the whole enzyme world globally has used the JEFA specification so far. And this was also sufficient to support the safety of our products also in the EU, in France and Denmark. It is really, it would really for the industry a big topic to introduce a new method, to change all the documentation, to have different specs around the world, to produce 3 new batches for products that may not be produced so often. So, and also this seems not to be really a safety issue, as our products have been safe, and also not all Enterobacteriaceae are pathogens. So we consider that the change is not really proportionate. And we hope that you can take into account our comments.
Yes, Sandro, you want to come back?
Yeah, this is a problem that we can discuss it again before the formal adoption, for sure. It is not a big change in terms of methodology, because it's almost the same medium, just sugar which is different for the detection. It is an ISO method where it is a standard analysis that is made in each single lab. So I think that enterobacteriaceae provide a better solution, but I would leave also to Livesh is also here to comment as well.
Indeed, in fact, we wanted to simplify to simplify it because we think the detection of pathogens is quite more expensive than the detection of an indicator and less relevant because it are the indicators which are important for hygiene measurement, and therefore we changed to the most optimal hygiene indicators at the moment, and that's E. coli and Enterobacteriaceae, because coliforms are phasing out in legislation, but also in practice, and that's because they are not so well taxonomically defined. But like Pierre Sandro said, we can discuss maybe the two options to keep it open and to open, but we have to discuss in the working group, of course, but it's not that the other indicators like coliforms are bad, but these new ones are more defined and are in that respect more optimal. But I think we can discuss your comment.
Yeah, okay, thanks a lot for your reflections. And I also understand your line of thinking. So I appreciate when you can reconsider or rediscuss. And if you would like to have more input from our side, we're very welcome to further discuss also.
And please, feel free to sort of contact the unit with arguments to the contrary. I think we were trying to simplify things here because we know from microbial enzymes we're essentially dealing with a product, a food enzyme, which has been so-called germ filtered, and where we really don't expect to have much in the way of microbial contamination, if any at all. So I think this sort of assay is a safeguard assay and nothing much else.
Yeah, I think the issue also lies a bit in the global application of our enzymes.
Well, I do totally appreciate what you say in terms of you're having to make, if you have a global market, you're having to make applications to other authorities. But I think we do have a fundamental difference in that we cannot just follow JEGFA, because the whole basis of the assessment used by JEGFA is fundamentally different from that the European Commission has chosen to follow. So we have the distinction between assessment of food enzyme preparations, which would be JEGFA, as opposed to the food enzyme, which would be the Commission. And the two are quite different. So we do have problems just following what JEGFA proposes, leaving aside other scientific differences, but that's another topic. So do we have other questions for the first part of this discussion?
No, there is no more chats. Everything has been answered, I would say.
Well, let's find out. Does anyone want to come in for the last 2 or 3 minutes? Now is your moment. If not, I'm going to exert my most important role as chair and declare it's time for coffee. And
that
so
is highly welcome.
And so we will break now until 4 o'clock, and then we will reconvene. Is that okay?
Okay.
So we'll
Thank you.
see you all back here at 4 o'clock.
Okay, bye.
Okay, welcome back, everybody. I hope we're, I think, ready to start our next part of this afternoon. And I'd like to introduce Professor Pier-Sandro Coccagelli, who is going to deal with the characteristics of food enzymes derived from microorganisms. And I would guess that this is probably the area in which we've had the most questions in the past. So I'm sure Pier-Sandra will have a number of issues to discuss with you. Pier-Sandra.
Thank you very much, Andrew. Good afternoon.
Sorry, yeah, I can't hear.
Could you hear me?
No.
Yes, but you are not yet in the presentation mode, Pier-Sandro.
Yeah, I should be in the presentation mode. Yeah, wait a minute. Is it now presentation mode?
Yes, now it's fine.
Yes,
Perfect.
thank you very much. Okay, so good afternoon. The aim of my presentation is
it
just...
only me who are having difficulty hearing, or is this a general problem?
I think it's you, Andrew.
No, it's you. We can hear perfectly well.
Yeah, we don't have a problem. Yeah.
Okay, thank you very much. So the aim is to present the section from 1.1 to 1.3 of the guidance, which focuses on the food enzymes produced by microorganisms. This presentation will mainly deals with the novelties that we have introduced since 2019, and in particular on the question raised during the open consultation. Let me start with a sort of timeline, because the guidance that we are discussing today is actually the result of a number of previous documents, starting from 2009, the first guidance on submission of dossier for food enzymes, but more importantly, in the last 3-4 years, the feed-up guidance, the first that would introduce the whole genome sequence, WGS, as a central action in the risk assessment of microbial products. Then the documents, the SEP statement that we discussed already together in 2019 on the characterization of microorganisms we use for food enzymes. And more recently, this year, the EFSA statement on the requirements for whole genome sequencing. So a document which contains all the criteria for whole genome sequence-based risk assessment. What's new? Actually, as I said, I will focus on what's new, and in particular, the just mentioned EFSA statement that provide the basis for the whole genome sequence analysis for the assessment of microorganisms intentionally introduced in the food chain, but also some improvement in the 2019 statement, in particular based on the experience of 2 years of application. For instance, we have deleted the request for phenotypic determination of antimicrobial resistance. We are taking consideration the rapidly evolving scientific methodologies, DNA sequencing and bioinformatics, and then, probably most importantly, your comments during public consultation. This is just a simple scheme that tries to summarize the different, let me take the laser, okay, the different step in WGS-based risk assessment, which is requested for bacteria and yeast strains, but is also recommended for filamentous fungi. So starting from the WGS analysis, we can reach an unequivocal taxonomical identification. We can look for gene of concern, and here we have 2 major categories: antimicrobial resistant genes, which is typical for bacteria, and toxigenicity and pathogenicity factors, and as well the determination of genetic modification. This is the central and probably quite critical point, the EFSA statement for whole genome sequence analysis that actually requests that the WGS raw data should be submitted to EFSA, and all the type of formats are accepted. Clearly, there are the raw data such as FASTQ in case of short-read sequencing, or BAM files in case of long-read sequences, and the assembled sequences in a FASTA format. Draft genomes are considered acceptable if provide sufficient information for an adequate characterization of the strain. So we don't require the complete and close genome sequence, but we need all the information that are necessary for an adequate risk assessment. Taxonomy. Taxonomy, just minor changes compared to the 2019 statement, and for bacteria and yeast should be totally based on genomics. In the case of bacteria, we have introduced two clear parameters, and in the case of the digital DNA hybridization, the 70% threshold is used to distinguish organisms belonging to the same species. And in the case of average nucleotide identity, we have taken the published and internationally recognized level of 95%. But we know sometimes that some of the species that do not fall some of the strain used for the production of the enzyme do not fall in a well already described species. In this case, phylogenomic can be used to identify the closest related species, which is fundamental for the risk assessment of a production strain. In the case of yeast, there are two options, or a phylogenomic analysis, or the alignment to a complete reference genome from the same species. For instance, in Pichia, there is a good reference genome, such as for Saccharomyces cerevisiae. Filamentous fungi, we have two options, with or without WGS. In the case of WGS, exactly the same approach used for yeast. When WGS is not available, the typical taxonomical approach, such as the sequence analysis of the ITS, internal transcribed region, or the 16S rDNA, are considered acceptable methods for the identification of filamentous fungi. When there is a contradiction on taxonomy, this should be documented, and a mutagenesis step should be reported also in case of non-GM, non-genetically modified organisms. Next step is 1.1.8 section, deals with the identification of genes of potential concern. There are two major categories: antimicrobial resistance, which is based on WGS, and it is the difference between intrinsic and acquired. I will spend just a few minutes on it because several questions were raised during the public consultation. And we have defined threshold values. Actually, we have used the threshold value that are defined in the EFSA statement of 2021, which is the 80% of identity and 70% of length. In case of homologies of this type in the database search, the hits should be reported. Gene of concern, toxigenicity and virulence factors. When WGS is available, database search, same parameters for reporting the homologies. When, in the case of filamentous fungi, no WGS data are available, a literature search is requested, as was in the 2018 statement of SEP panel. Antimicrobial resistance, some more detail, at least two updated databases. The threshold values that we already described, 80% of identity at protein or nucleotide level, depending to the database, and 70% of coverage with the subject sequence. This is a new point. When two or more fragments covering less than 70% length of the subject sequence, but with at least 80% of identity, it means that a portion of a gene is on a contig and the other on another contig. In this case, it is requested to check if the full gene is present. So could happen is a rare situation in which half of the gene is in a contig and the remaining part is another contig. So we request to analyze better this situation. When an antimicrobial resistant genes, and in particular an acquired gene, is present, there is a practical consequence, and demonstration of absence of viable cells and DNA of the production strain in the food enzyme should be provided. Few words about intrinsic versus acquired. It is a pretty old approach. I think it was used the first time in 2005 in EFSA. For the, if I remember well, for the feed additives. And acquired resistance is when a strain of a typically susceptible species is resistant to a given antimicrobial drug and differs from intrinsic resistance, which is inherent to a bacterial species and is typical of all the strains of that species. And intrinsic antimicrobial resistance is generally not considered a safety concern. Just a very simple example, Vancomycin resistance in enterofermentative lactobacilluses and in Enterococcus. In the first case, the mechanism of action is exactly the same. There's just a substitution of one amino acid with the lactic acid in the cell wall precursor. But in all the heterofermentative lactobacilli, such as Lactobacillus plantarum, all the strains are resistant to vancomycin. Differently, in Enterococcus, only the strain that acquired by horizontal gene transfer, the DILAC ligase, are resistant and therefore are considered, this acquisition is considered a risk. QPS, Lieve Herman will present later on more details on QPS, but when we talk about toxigenicity and pathogenicity, QPS strains are considered toxicological safe. So no other information is required other than the qualification, such as in the case of Bacillus and related genera, or in the case of GM, where the QPS is parental, the QPS concept is also applicable to the GM unless the genetic modification has introduced a new safety concern. When the strain is not QPS, the situation is different between bacteria and eukaryotes. In case of bacteria, the search focuses on the known virulence factors, which are present in specific databases. In the case of eukaryotes, if WGS is available, the same, the search for gene encoding, for instance, toxin or toxin production pathways, such as in the case of mycotoxin in filamentous fungi. When WGS is not available, the literature search to identify the capacity of the species of or closely related species to produce known toxic compounds. Genetic modification, nothing new, exactly the same approach. When WGS is available, is the comparison with the parental strain. So unmodified strain with the production strain, focusing on what new has been introduced, and for each of the new introduced DNA fragment, it should be reported the origin, the function, the intended effect and gene of concern, as well as the deletion or the modification in minor modifications, such as changing in the codons, such as codon optimization, should be reported. When WGS is not available, it is the traditional approach before 2018. So all the steps should be described with each single step, the clear documentation of the genes that have been introduced, in particular taking consideration plasmid, gene of concern such as antimicrobial resistant genes. Well, and last, with no changes to the current SEP statement, is the viable cells, the absence of viable cells of the production strain in the product. Nothing has been changed. 1 gram or 1 ml of product, 9 samples from at least 3 batches, and all the appropriate controls. And the same applies to the detection of DNA of the production strain. It is requested for GM production strain or non-GM production strain with acquired antimicrobial resistant genes. It is a PCR methodology, and the threshold value, the DNA should not be detected in a PCR approach that has a limit of detection of 10 nanogram of control DNA or lower. So this is the situation, so no new changes. So I thank you for your attention, and I wait for your comments at the end of this session.
Thank you very much, Pier-Sandro. I think I will stress that all of the data that's in the latest statement from EFSA is also incorporated into our guidance. And so you should find all of the information. As we said, the guidance is intended as a standalone document. So you should find all of the information, all the data requirements in the guidance. And that should align with the statement. So now we move on to LEVA, and as Pier-Sandro indicated, the issue of QPS and when we can waive the need for toxicological studies. LEVA, I pass the floor to you.
Yeah, thank you, Andrew. I'm bringing the good news. Toxicological studies are not always needed, but only for some specific cases and only for certain conditions. So toxicological studies, they might be waived for food enzymes, which are derived from edible parts of the plants and animals, and this in relation to their dietary exposure. So this dietary exposure to the food enzyme TOS should be within the same magnitude as the dietary intake of the fraction of the plant or animal material comparable to the food enzyme TOS. This dietary requirement is not necessary for animal rennet, and that was an answer of one of your questions, because these enzymes have a long history of safe and wide consumption. So toxicological studies might also be waived when it can be demonstrated that there is no or negligible carryover of the food enzyme TOS in the final food product. And I give here some examples which we have seen already in some applications. This is the case in the distillation processes, starch processing. And of course, I understand the question. Please explain what we mean with negligible carryover. And in our answer, we refer to two parts of the guidance. So to the annex, where we say that more than 99% of removal is acceptable. And we also refer to section 5.2 of the guidance on the removal and the absence of TOS during the manufacturing process, a part that our colleague Jose already overviewed. Okay, toxicological studies might also be waived when your enzyme is produced from a microorganism which belongs to a species which qualifies for the presumption of safety, so a QPS organism. And also in that case, but my colleague Pier-Sandro mentioned it already, in that case also no search of the whole genome sequence for virulence and pathogenicity genes is required. QPS, the qualified presumption of safety, provides a generic safety status for microorganisms intentionally used in the food and feed chains. So let it be clear, because it was also a question, that it's not for plants or products derived from plant origin. On the contrary, and this was also a question, QPS includes also protists and microalgae, and because QPS is also extended to genetically modified organisms, this is also extended to protists and microalgae. So you can have GM microalgae on the QPS or considered as a QPS, which can follow the QPS assessment and so the waiving of the tox studies. I give you here the 3 QPS species which are at the moment already on the QPS list. It's Euglena gracilis, Aurantiochytrium limacinum, and Tetraselmis huey. A very important aspect is that QPS provides a safety statement at the species level. So all strains belonging to that QPS species are following the QPS assessment. So to respond to a question, so there is in fact no danger to consumers due to intra-species diversity in between a QPS species. Yeah. But, and here is where again this but, even for food enzymes produced by QPS organism, the application, so we need some information on the microorganisms. So the tox studies can be waived, but we need a very good taxonomic identification of the microorganism, like Pier Sandro already explained, preferentially based on whole genome sequence. And this taxonomic identification is in fact the basis, because it's the proof that your strain indeed can follow the QPS assessment. The safety of the genetic modification, if it's a GM strain, needs to be confirmed. And also, and that's a very important one, check on the QPS list the asterisks to look which qualifications should be documented in the dossiers, because a lot of QPS organisms, QPS species are QPS just with a qualification. And I explain in the next slide. What's a qualification? A qualification is a potential hazard which have a potential hazard which can exist in some strains of the QPS species. So it's a hazard which has to be tested at the strain level. So in the dossier that you send, it will be clear when I give you 2 examples. A generic qualification for all QPS bacterial strains is that antimicrobial resistant genes need to be absent. And in the QPS document, and also for answering a question on that, it's clearly stated that it's the absence of acquired genes conferring resistance to clinically relevant antimicrobials. So these genes need to be confirmed in the dossier to be absent. Another qualification is for the production organisms. So you can have a QPS species only for production purposes. This is, for example, the case for some yeast species which are used in your applications, Candida and Comagatella and Pichia. And in this case, you need to confirm in the dossier, even for this QPS organisms, that there are no viable cells in the product present. Yes. A very common question, which is very often asked, is how can I now get my strain on the QPS list? And unfortunately, I have to disappoint you on this aspect. At the moment, it's not possible to know in advance before sending a dossier to EFSA that your organism can come on the QPS list. It's only for those species which are already on the QPS list that you can be sure. But for a new organism which is not yet on the list, the assessment only starts when the dossier, the notification comes or arrived at EFSA. So there is at the moment no two-step process available to know it in advance. Yes. Toxicological studies might also be waived for non-QPS microorganisms. And the first one is a very special case because, in fact, that's the case when your strain belongs to the QPS list. It's the same species, so your strain has the species of the QPS list, but it contains an antimicrobial resistant gene. An important qualification, which means that in fact your strain is strictly, in strict sense, no QPS because the qualification is not met. But the tox studies can anyway be waived if you can carefully show, and Pier Sandro explained how you have to do it, if you provide us the documentation that no DNA of this EMR gene is present in the product. Special case. The more normal case that we see is for the non-QPS organisms. So, for example, the filamentous fungi. Also there is a possibility that the tox studies can be waived. And in fact, in that case, that a substitutive set of tox data can be used when, and these are coming then from a strain which is very related to the production strain which is under assessment. And this is a very careful evaluation, not an easy one, on a case-by-case basis. And I explain in the next slide. Because this substitutive data set has to be produced, like I said, from a very well related strain. And in the guidance, we say of the same strain lineage, which in fact means derived from a common progenitor. No conventional mutagenesis steps between the two different production strains have been carried out because conventional mutagenesis can have consequences for the phenotype. And then the third one, also the genetic modification should be compared very carefully, and therefore also in this case, so also here for fungal strains, we need the whole genome sequence, because it's only with the whole genome sequence assessment that we can look, that we can be sure that no harmful components would be induced between the two strains, the one we used the tox studies from and the one under assessment. And then in my last slide, just a very general comment. Of course, always, also when all the tox studies are waived, we have to be ensured that the manufacturing process is sufficiently under control, that there are no extra concerns coming in the product because of the manufacturing process, and that the enzyme is sufficiently purified. And in my last slide, I give you the links to the QPS list and also to some documents on the QPS topic. And I will be happy to answer your questions in the discussion later on. Thank you very much.
Thank you, Lieve. That was very clear. Yes, unfortunately, I think questions are going to have to wait until that discussion period later. We now have three very quick and quite different presentations, one after another. and I apologize for this, but there are elements we do need to consider. So we go from genotoxicity to allogenicity to dietary exposure in half an hour. So the first of these, of course, is the issue of genotoxicity testing and the strategy that's used for food enzymes, and I have to say across most of EFSA. And Professor Claudia Bolognesi will introduce you.
Thank you, Andrew. I will try to summarize the main issue for the evaluation of genotoxicity for food enzyme. The food enzyme are a complex mixture of unidentified components, except for the declared enzymes. And from the EFSA statement on genotoxicity assessment on chemical mixture, the testing strategy for the food enzyme of yeast fraction should follow the scientific committee testing strategy guidance for individual chemical substances. The next slide report this strategy. The EFSA strategy for the genotoxicity evaluation of substances in food and feed involve a stepwise approach. This stepwise approach starts with the first tier that involve the evaluation of genotoxicity in vitro, including two tests, the bacteria reverse mutation assay and the mammalian micronucleus test, involving the evaluation of the three genetic endpoints for genotoxicity. In case for food enzyme, in case the Ames test is not applicable, a test for induction of gene mutation can also be applied but need to be justified. The next slide. In the application of bacterial reverse mutation, the application of treat and plate assay or treat and wash assay is recommended in order to avoid the feed effect due to the presence of histidine or tryptophan in the medium. The most important recommendation is the range of concentration to be applied. The recommended maximum concentration for soluble non-cytotoxic substances is at least 5 mg TOS per plate. The concentration needs to be calculated and expressed as TOS. And in cases this concentration is not toxic, higher concentration can also be applied. The next slide. The next slide is the mammalian cell micronucleus test. The recommendation is to apply the cytokerasin B as a cytokinesis blocker in order to obtain a sound result with respect to the frequencies of micronuclei. Again, the range of concentration. In this case, the maximum concentration needs to be based on cytotoxicity. And in cases of precipitate, in absence of precipitate or cytotoxicity, the maximum concentration may be higher with respect to the limit of 10 millimolar or 2 mg per milliliter reported in the OECD guidelines, in order to increase the concentration of the minor component of the mixture and to have the possibility to detect also minor genotoxic compound in the mixture. Next slide. The results. If you obtain negative results with both the test, no further testing is needed, and the concern for genotoxicity can be ruled out. In case of positive result, the recommendation is to review these results on the basis of the data available. In cases of Ames test, the factors to be considered are the toxicity, to see if the level of toxicity producing a positive result in terms of increase of revertant is too high. The enzyme reactivity, in order to test if toxic compound may be released in the medium. And finally, the analytical data to test the presence of histidine or tryptophan. After this analysis, further testing could be established, considering different concentration range, different methods, or the use of inactivated enzyme. In case of positive result with the micronucleus test, the factor to be considered include again the toxicity for sure of the enzyme reactivity, but also the scoring of new slides. Because in the micronucleus test, we can save and store the slide for further analysis. And another possibility is to test, to investigate the mechanism for the micronucleus induction. Performing the fluorescence in situ hybridization with the DNA probes of crest staining, we can distinguish between clastogenic and angiogenic mixture. In this case, we have more elements, more data, in order to address the in vivo follow-up. Next slide. For the in vivo follow-up, we need to consider the genotoxic endpoint which provide a positive result in vitro. In case of a follow-up of in vitro positive result with the Ames test for gene mutation in bacteria, the most appropriate test to be conducted is the transgenic gene mutation assay or the in vivo mammalian alkaline comet assay, considering the evaluation of different target tissues with comet assay. For positive compound detected with micronucleus in mammalian cell, we can distinguish between clastogenic mixture and angiogenic mixture. For clastogenic, we can use a combined test, including the mammalian erythrocyte micronucleus test and the mammalian alkaline comet assay, considering the stomach, to test the stomach if you have positive result in vitro without metabolic activation, or liver if the result were obtained with metabolic activation. In cases of angiogenic effect detected in vitro, we have only one test for which is available OECD guidelines, and this test is the mammalian erythrocyte micronucleus test. But nowadays, the experimental protocol for the evaluation of micronuclei in liver and also in gastrointestinal tract has been established. And for the micronucleus in liver, OECD guidelines is in preparation. And for micronucleus in gastrointestinal tract, the validation of the test is ongoing. So we have the possibility to combine this evaluation, the evaluation of micronuclei in liver, for in vitro positive with S9, or in gastrointestinal tract for in vitro positive without S9. Also in this case, we need to consider the range of doses. The highest dose to be tested need to be the maximum tolerated dose in animals treated. And if in the dose ranging finding test we didn't obtain toxic result, higher doses than the maximum limit in the OECD test guidelines can be considered, such as more than 2000 milligrams per kilogram body weight. The highest dose to be applied should be limited only by the maximum volume that should be given to rodents. That is reported in the OECD guidelines. That is 1 milliliter for 100 gram body weight, or 2 milliliters for 100 gram for aqueous solution. This is a new consideration come from a discussion in the Genotoxicity working group, and this recommendation will be introduced in the final version of the guidance for food enzyme. Next slide, please. What about the result for the genotoxicity assessment for the in vivo follow-up? If the in vivo testing of an in vitro positive mixture provides negative results, the relevance of this finding obtained with in vivo follow-up will depend on the genetic effect assessed. We need to consider the genetic effect producing positive result in vitro, the test protocol applied, the route of exposure, and mainly the tissue to be considered, the expert judgment on the reliability of the result obtained, including the consideration of the target tissue exposure. The target tissue exposure is a crucial question because the assessment of systemic genotoxic effect in liver or bone marrow may be limited by the fact that the target tissue exposure cannot be demonstrated, as any toxic effect elicited in the target tissue by the mixture cannot be unequivocally attributed to the in vitro genotoxic component, because we don't know which is the component inducing the genotoxic effect in vitro. Next, in the next slide, there are reported the line of evidence for bone marrow exposure reported in the EFSA opinion on clarification of some aspects related to genotoxicity assessment. In the case of mixture, we have only 4 lines of evidence to be considered to detect the bone marrow exposure. The toxicity to the bone marrow in the mammalian erythrocyte micronucleus test, that is the decrease in polychromatic erythrocyte with respect to the total erythrocyte, and the toxicity to the bone marrow observed, for example, in a toxicity study, in this case the 90-day studies, which was conducted in parallel with the same batch of the food enzyme. And again, we can check the systemic toxicity, which is observed in the bone marrow micronucleus test or in the systemic toxicity observed in toxicity studies. The next slide is the last slide for me, report the list of the EFSA guidance relevant to the evaluation of genotoxicity for the food enzyme, and include 3 published guidance. And the last one is the guidance on allogenicity, which will be is already finalized and will be published in a few months. Thank you for the attention, and I'm ready to answer possible questions.
Thank you very much, Claudia. Oops. So we have a very rapid change of topic, I'm afraid. We move from genotoxicity to allogenicity. And I'd like to introduce Professor Henk van Leeuwen, who will deal with this topic and who is the working group expert on allogenicity. Henk, it's over to you.
Yes, thank you very much. Can you hear me?
Yes, Henk, we can hear you clearly, but you are not in the presentation mode yet.
I thought I was. Can you see my screen?
Yes.
Yes, and the presentation.
Fine now, okay.
Okay, thank you. Okay. So what I'll do first is to shortly rehearse what are the requirements for allogenicity assessment of enzymes. The assessment of potential allogenicity considers A, the source of the food enzyme, B, the declared enzyme or enzymes, and C, any proteinaceous material with known allergenic properties included in the fermentation medium, if that is the case. Quantifying the risk of allergenicity is currently not considered possible in view of the varying individual susceptibility to food allergens. Even trace amounts of allergic protein transferred to the food could potentially present a hazard for sensitized individuals. And a lack of transfer will only be accepted if it can be demonstrated that the food enzyme TOS is absent in the final food. And for all other cases, the following information is required. First, investigation of the amino acid sequence of the food enzyme, and any sequence identity greater than 35% to a known allergen using a sliding window of 80 amino acids should be reported. And hits found should be discussed in the context of the available literature information. And then a search of the potential of the literature of the potential sensitization or elicitation reactions by the specific enzymes in the application. And the route of exposure should be presented, for instance, the assessment of safety at the workplace, such as sensitization studies that are there very often, respiratory sensitization. They should be submitted. And finally, a search for possible allergic reactions caused by the enzymes of the same family of enzymes as the one under application should be performed. And in case allergic reactions have been observed, in such cases, a rationale should be given on how these observations do or do not impact the evaluation. And what I will now do is, I just have a few very short lines that sort of capture the questions we had in the public consultation. And first, and I think the most pronounced question was, is this 35 cutoff of sequence homology using a rolling window of 80 amino acids, is that not too conservative? Does that not lead to many false positives? And the answer is yes, it does lead to false positives. And the question was also, should it not be raised to 50%? So yes, you will definitely have less false positives at the cost, of course, of having more false negatives. I'd like to say that the requirement now is very much in line with other guidances as they exist in EFSA and also with FAO. I'd like to say that even if sequence homology is found with a known allergen, that does not lead automatically to saying that this enzyme is problematic in terms of food allergy. Depends also very much on the type of homology that you see. For instance, if there's homology with a respiratory allergen known in the occupational, very often in the occupational setting, it's known that people respiratory sensitized to occupational allergens can ingest quite a lot of allergen without facing any problems. Also homology, for instance, with some outlandish allergens such as snake venom, for instance. We have these cases that doesn't lead to many questions. So in the end, what you will see, even if there is homologies, and that happens, I wouldn't say always, but that happens to a certain extent, then in most cases, still at the end of the day, the likelihood is deemed very low. Only in cases when there's homology with a known food allergen, then the situation may be a little bit different. And then the conclusion may be that yes, there may be reactions expected. Also, the same is maybe true for those allergens that share homology with allergens that are associated with the food allergy, with the oral allergy syndrome, which is associated with pollen allergens. So in those cases, that may be a little bit different. And then, but these can be refuted quite easily if IgE binding studies or even skin prick testing, but let's stay with IgE binding studies are performed. So in case the applicant wants to negate such findings, then this can be done. We don't require this as a default. So that was another question. Should we not always do IgE binding studies? And the answer is no. We think that's quite an overkill in view of the fact that not many food enzymes will be identified as potential allergens, first of all. And if there is a sequence homology to a certain allergen, very often the specific anti-sera, IgE sera, are not even available. So we don't ask for this, although obviously if the applicant wants to do it and has the capacity to do it, he is allowed to do it and show the information to us. Bit different issue is the question on 6 to 9 or 6 to 10 amino acids. Would the sliding window of such a small sequence not be more appropriate? Because these constitute the epitopes to which the immune system reacts. And currently, doing this kind of exercise is not more predictive, not better predictive than the 35 cutoff with the 80 amino acid sequence. This has not been accepted currently. There is some value in doing this. We won't deny it, but we don't request this as a default. It may especially have some bearing on prediction of proteins that could induce celiac disease, but I'll come to that later. What about 3D analysis? Because the structure of the protein, the 3D structure is also very decisive, obviously, in the end of the allogenicity. And we acknowledge that quite a lot of work is being done on this, and there is a lot of progress. And perhaps in the end, in silico analysis, using 3D analysis, using the epitopes, the 9 to 10 amino acid epitopes, the sequence homology for the whole protein may actually be leading to a better prediction of allogenicity, and in particular food allogenicity, but we are nowhere there, and that needs further research. There was a meeting from the GMO panel the other week, 1 week ago, on this, and that was clearly stated there. So we're not asking currently for 3D analysis. Yeah, I already said something about celiac disease. What about proteins inducing celiac disease? Should we not also look for that? And there, the 9 amino acid, the motifs that in gluten that produce celiac disease are well known, and motifs that are very much looking alike that may in the end also be effective in causing a celiac disease or having reactions in celiac disease patients. But it should also be said that people that are hypersensitive to gluten can ingest quite a lot of proteins that are associated with this disease without acquiring the negative effects, and the doses that they will get are certainly beyond what we expect with the food enzymes. So we're not requesting this issue. Then there was the question, why referring enzymes, two enzymes from the same family of enzymes, even if the proteins may be different? And it's true, proteins may have the same enzymatic activity and yet be so different that in allogenicity terms they are quite different. That's very true. But the reverse may also be true, that there are enzymes that are a bit more similar. And of course, looking at the 35% homology cutoff, that is only as good as the databases on which this whole thing is based are valid. And they are quite good, but they're also still developing. And so it's not 100% full proof that everything is there. So for that reason, we ask for a literature search, not wet research, but for a literature search looking at enzymes from the same family. What about why indicating raw materials in the culture medium? And that question was there because already the scientific committee earlier on had indicated that the raw materials in culture medium of microorganisms that produce the enzymes these raw materials will be consumed and will not pose a problem. And that is true, and the panel agrees to that. And the only reason to have this is for completeness. According to the regulation for food production, any food produced with an allergenic source or an allergenic component needs to be labeled as long as there is as long as it has to do with allergens that are in the Annex 2, which, for instance, soy or egg or something else. And so that is here. But basically, it's very true, and that is actually all the time we come to the same conclusion, that even if these raw materials are in the culture medium, at the end, we don't see a risk. The only time we may see a risk is when there is a downstream application of an allergenic moiety in that medium, such as, for instance, lysozyme. We had a few cases of that. Then there was a question, what about adjuvanticity? An adjuvant in immunologic terms is usually based used as a term for vaccines to enhance the efficacy of vaccines. But the more broader term, of course, can also be taken as inadvertent stimulation of the immune system, including inadvertent stimulation of allergic responses, which is an immunologic response. However, this is not allogenicity itself. An adjuvant is not necessarily, it could be, but it's not necessarily an allergen. The well-known cases, diesel exhaust is adjuvant for pollen allergy, but it's definitely not an allergen. So we don't feel that belongs to the realm of allergenicity. It belongs to the realm of toxicity to the immune system, and that is being dealt with in the toxicological section. I have to say that the immune parameters in the toxicological section are not very major, but in any case, the immune system to a certain extent is looked at in the general toxicity section. So that is covered, but not under the realm of allogenicity. And I believe that is my last slide. Yes, thank you very much.
Thank you very much, Henk. I'm sorry we move again to a completely different topic and to the final part of both of our opinions and the guidance, the final part, but not the least part, and that's exposure assessment. It's unfortunate that the expert on the working group who predominantly deals with exposure assessment couldn't be with us today, Christina Torres. But E has volunteered to take her place. So E, I open the floor to you.
Thank you, Chair. Thank you, Andrew. And yes, and I'm EFSA staff, and I'm here basically fill up the gap the experts Christina Touloustus this moment has created, and so I hope I am up to her standard. And next slide, please. I think before going to the comments, and I would like to make a recap of what has been extensively clarified in March. And so first of all, the dietary exposure assessment and the very end consumer is actually the European consumer. And for this reason, and our consumption data are very much primarily based in the consumption data collected on European populations. And of course, we are using the actual consumption data, and but this does not mean that we are moving away from the conservative scenario. We do, and we try to strike between the balance of being conservative to protect the safety of the consumer at the same time and be, let's say, actual. So to avoid a super overestimate or ungrounded, let's say, attribute values or factors and so on. And the second important message is actually the dietary exposure is always carried out for every dossier we have assessed, and but that does not mean that for every dossier it's necessary to compute the intake to the food enzyme TOS. And I think this is very much an outcome of the use of the enzyme itself during the different food manufacturing processes. And for this reason, and we have decided that if there is evidence to prove that there is no residue, food enzyme TOS residue transferred into the final foods as consumed, and there is no need to make any effort for any computation. And of course, this shifts the burden to prove the absence of the residue. And thirdly, the entire dietary exposure assessment and irrespective of whether the computation step takes place or not. And the food manufacturing process in which the food enzyme is used is actually serves as a guiding binary and to the whole evaluation. And so when a calculation is deemed necessary, and this guiding principle would need to anchor on two very critical input factors. One is the relevant food groups, and we translate that and into the so-called food X categories. And secondly is different technical factors, be it ingredients factors or be it simply a conversion between different commodity and add the food, the what we call the raw material or substrate, some people would like to call it in that way, and to which the food enzyme is physically applied to. And in some occasions, for some niche foods, to avoid over calculation, and we also introduced a so-called food occurrence factor, and that's F3 factor for the one that is had a close look on that. And in order to ensure that EFSA really gets this part of the expertise right, and we put out our intention of which food we would like to consider, which factor we would like to consider for each foods, and into a big Excel spreadsheets. And we put that to the open public and to collect the feedback, because we believe that the food manufacturer is actually the one and the know-how. And of course, with this said, and I must also say that EFSA has not yet finished, let's say, combing through this so-called different food manufacturing processes. So more call for data and are being launched. And currently there is one about lactose degradation is on the web and looking for your input. And with this situation explained, and of course, I also need to clarify that currently there are 6 single food process FIME calculators available for you to use. And these calculators can be used by you to assemble your dossier, and also for anybody that is interested to know at which magnitude and I might really get exposed and to the food enzyme toss. And in the case of a food enzyme would be employed in different or more than 1, 2 and above food processes, and EFSA carries out the calculation. And this is because EFSA is let's say, we have the direct access and to the individual consumption data, but not only that's the only reason. A second reason is when there are, let's assume, and 2, one enzyme will be used into 2 different food processes, and we would actually need to come up with a third consideration, that is which food is more dominant and how am I going to use one factor. And just think about you are eating a cake and the enzyme comes from the egg components, also come from the flour components, and in that case, and how are we going to compute? And it's not a simple sum up, because a sum up would absolutely overestimate. And so for those mathematical questions, and currently, and our statisticians in-house and working with us and to solve this. But of course, when all this is done, and one web form calculator will be available, and that form will be capable of dealing with multiple food processes. And next slide, please. With that summary made, and I will draw your attention to the comments collected during the public consultation. And I have to say that the dietary exposure had the easy part, not so many comments have been collected. and they are technical. So the first one is about the comparable fraction. And the second one is, of course, and if it's not a food manufacture process is not covered by the Commission working document, and what are we going to do? Legitimate question. And then there are two questions very much focused on how to demonstrate the removal. And the next slide, please. And so to explain the comparable fraction, and is not possible to make it impossible to make it clear unless I show you these two sets of estimation side by side. And so essentially, if you are familiar or if you have already attended the webinar in March and you understand that for enzymes extracted from the edible plant parts, and we carry out two sets of dietary intake estimate, and in order to put this, let's say, legal term consumed in a comparable way and into the risk assessment parameter or outcome in this case. And so the comparable fraction of the source material. So you look at it, source material, so you know that that means the physical material and to which the enzyme is extracted, and comparable. So the comparable essentially is a yield factor. So if you look at the red highlighted text, and that is really what it's about. And in a simple mathematical term, and it actually refers to 1 kilogram of food enzyme, and typically need to be obtained from, for example, 1,000, and a kilogram of the vegetable peels. I put example here because we have the Cucurbita opinion published. And if you go into that opinion, I think this comparable fraction is quite well illustrated, and also this so-called yield factor is quite well illustrated there. So it's actually one factor embedded in the calculation sheets. Next slide, please. And okay, so new food application. And I think here, the real question is who should be doing what? And I would say that for a new food application, and when you prepare the dossier, and of course, and is the applicant's responsibility really to provide the technical descriptions and very detailed. And this includes a diagram and illustrate how the food is really produced and put the enzyme into its context. And in case there are steps of removal, separation, and in which the food enzyme toss can be removed, and that you really have to mark it and explain it in terms of which technology, how, and how much is your assumed or your assumption. And in case there is no such removal process, is simply the enzyme just inactivated and then remain in the final foods. And I think for that, and also we would really need you and to try to think in a dietary exposure terms, and try to provide as exhaustive as you can. For example, what kind of final foods this enzyme may end. And also, if you are deep into dietary exposure, you can even give us certain factors, and that is definitely welcome. So I think the guiding principle is try to put yourself into a dietary exposure shoes and assemble this part of the dossier. And when the dossier arrives EFSA, and how we do, and of course we will treat this new food application and as what we have known and as what we have seen. And that is why that I put this diagram that I have explained very exhaustively and 3 months ago. And this is just to say that EFSA, we will follow this diagram, and starting to look whether it's necessary to carry out the estimation or not. And if no, and we will focus on the evidence of the removal. And in this case, for these kind of new food applications, and I'm sorry, I think this burden of proof will be very much, I think, bared by the individual applicant. And this will be a different situation and than what you have seen so far, that it's actually the industry association has been really providing all the theoretical calculation and analytical data. And the reason for that to happen, and this is because those processes is relevant to many applications and concerns many applicants. And so, of course, we can make use of the power of the association. But when it comes to very niche, very innovative food applications, and yeah, you should expect to receive further clarification requests from EFSA. Otherwise, and the rest of the work in terms of in case estimation needs to be carried out, and the EFSA will work with you and through the question and answers and under that application, and work the whole way through all the steps that is highlighted and in this diagram. So for the sake of time, and I'm not going to explain it again. And the next slide again, please. Okay, so what about this removal and absence? And there is one question, and there is one suggestion, and to say that, well, in our draft guidance, we use the word strong experimental data, of course, and we are asked, EFSA, please qualify what means strong. And so now we're going to replace it and with another word, and robust. Does this say a bit more? And I hope so for chemists. And so in order to explain that a bit better, and I made a very simple diagram. And let's say that follow this diagram and I will explain. And so this is a case that you look, there are two dashed lines and indicating that the food enzyme TOS is actually separated and further removed and by repeated washing. And so what I call that waste fraction, and that's where the food enzyme go, right? You trace it. And so this is so-called robust experimental data basically says several things. First of all, to prove that it's removed. And you could go with the indirect data in this case. So analyze the waste fraction. And but equally, and you can also analyze, use direct experimental evidence. That means follow the straight line and analyze either the blue circles or the pink circles. And perhaps analyze multiple steps is a good way. And so these are about analytical data. And of course, the analytical data is just empirical observations and what needs to be done. And I think those need to be done. And by putting it into the actually your assumption, your hypothesis, and that is a theoretical calculation, why you believe it's removed and to which extent it's removed. And so far, EFSA accepts that more than 99% of removal. And last but not least is the second bullet point is about the data itself. So as here we are talking about the food enzyme in the industrial food manufacturing process. So the generation of the data, the samples should be very ideally come from the industrial scale. And not take an Eppendorf, put 100 microliter of oil. No, that's really not okay. And I think with this, and I actually finished the presentation, and I'm looking forward to your questions. Thank you very much. Andrew, back to you.
Thank you very much, Yi. So now we're at the stage where we can accept questions on any of the areas we've discussed. I would suggest we sort of divide these in two and perhaps take any questions relating to the microbial issues, so microbial characterization of the food enzyme from microorganisms. This has been discussed many times before, but in case there are additional questions, and Liv Herman's presentation, particularly on the QPS elements. So I don't know whether we have any questions. Anyone who would like to raise a question in that area? Do we have any chat?
Yes, yes, we do. Yes, we do. I would first of all invite Nathalie Henning.
Nathalie? Hello? No?
Perhaps in this case, maybe Andrew, I could read out the question because I think it's interesting for the others.
Okay.
Yeah. So the question is, would it be possible to submit one subchronic toxicity study and for two enzymes produced by the same parental strain, for example, filamentous fungi? And however, the two production strain has undergone different mutagenesis steps. And the question is, can we do that? And also, would it be possible to only compare the known sequences for the biosynthesis of those secondary metabolites or mycotoxin?
Yes, Andrew.
I think that is more liver topics, but
okay.
Regarding presentation.
So I think he's passing the buck to you.
Andrew, I start answering, or
yes, please.
Yeah.
So Piers Andrew's just passed it over to you.
Yeah. Okay. So indeed, there is some question in relation to just conventional mutagenesis, and that we indeed exclude the possibility of waiving stock studies when there is a conventional mutagenesis step occurred between the two different strains. So the strain from which we have the tox studies and the strain which is under assessment. We are not accepting that. And that's because, like for non-QPS organisms, like filamentous fungi, conventional mutagenesis is introducing mutations very randomly in the genome. It can be insertions, it can be deletions, it can be point mutations, they can be quite extensively. And we, the main importance is that they are unpredicted. They are random, unpredicted. So at the moment, we have not sufficient knowledge on what an organism is producing and how all these components are interacting to, in fact, to base the safety just only on data of the sequence. We really need to rely on the toxicity studies and in vitro and in vivo toxicity studies. So we can't therefore not accept these conventional mutagenesis steps occurring between the two strains of comparison. And in fact, only the mycotoxins is not sufficient, like you asked, because there are also other metabolites involved in safety. There are other secondary metabolites from which we not know everything already. And there are also other components, like, for example, proteinases, which can be affected, proteinases which need in the other products which degrade maybe possible hazardous components. And if then a mutagenesis step is deleting the activity of this proteinase, also that can affect. So it's not only the induction of expression, it's also sometimes the deletion or the inactivating of some genes. So it's too complex to assess the safety of that just based on sequence data.
E, could you just confirm the question? Did the question mention 2 different enzyme activities from the same strain?
No, it was from 2 strains deriving from the same parental with different mutagenesis.
Oh, okay, then it's clear. I mean, I think we have a rule of thumb that says new strain, new application.
Yeah.
Okay. So, E, anything else on the microbial front? I
Yes.
see no hands raised. So.
Yes, look,
Okay.
I think if Marianne Dessen is in the audience, I would invite her to speak up.
Yes, I am in. Good afternoon, everyone. I have a question regarding the definition of the intrinsic resistance versus acquired resistance. So as it is described in the guidance, basically the definition says that we have to show that all strains from a species should carry the antimicrobial resistance to comply with this definition. For us, it's maybe a risk that this would lead to many strains being considered as a harboring acquired resistance, which of course is being seen as a hazard by EFSA, as we have now understood, and obviously increasing the burden to the applicant to show that there is no risk at the end. And the question was whether there would be, more of an understanding that basically not all the strains should be demonstrated to carry this resistance, but I would say many of the strains belonging to the species. Would that also be something that you would accept and consider?
Pier-Sandro, this is
yeah,
your area.
I can answer. Generally, when in a population there is the bimodal distribution of the MIC, so the value for resistance, it is considered that there are 2 different populations. So one is susceptible and the other one is resistant. So the fact that we say that all recognize the fact that is typical also of clinical microbiologists, which I'm not actually, in which the antimicrobial resistance is acquired. Acquisition can derive by mutation, by gene transfer. There are different cases, but in general, the impact on the food enzyme producer is the presence or absence of DNA. So if it's not a GMO organism and not genetically modified, and it is resistant to one of the antimicrobials that we have enlisted because all that has been found on the basis. We focus on critically important antimicrobials or highly important antimicrobials as defined by WHO. So if it's an antimicrobial, sometimes it's possible to identify genes coding for antimicrobials that are not used in clinical therapy. So this is not generally an issue. But if there is a gene which is not inherent in that species, that means that there are some strains that are lacking of that particular gene, we consider it a risk. So the absence of DNA should be demonstrated in the final product.
Does that answer your question?
Yeah, more or less. But I mean, what would be the difference? Again, I mean, maybe what would be the difference? Let's assume that we would have like 50 strains of a specific species that would be, carrying this. And what would be the difference between the case where 48 would basically carry this and the difference? Yeah, I mean, they are spreading anyway. So what you would like to?
No, it's not. I think that intrinsic resistances in most of the cases, and I did the case of vancomycin, taking really one of the paradigm of intrinsic resistance, are well described. So we know that groups. So it is not, most of the time, it's not even to the food enzyme producer to demonstrate that is intrinsic or it is acquired. There are enough publication and science behind to support that is intrinsic resistant or not.
Okay, good to me. Thank you.
Okay, thank you. E again.
Yes.
I have no hands raised, so it's. Yes, I do. I apologize. Okay.
No problem. Actually, I would like to invite Tronvo from Intertech. Is about non-QPS.
Oh, okay.
Hi everyone. So my question relates to the point on for food enzymes obtained from non-QPS production microorganisms, in which tox studies and other products from a very related microorganism may be used. I was wondering if EFSA has any criteria what would constitute very related microorganism, whether this is something that would be found in the scientific literature or done by some sort of other analysis.
Yes, I think it's very clear. It's very related what we mean. So it's the progenitor, so that we can have of that strain. So we need to know between the two strains which steps have occurred. So it's not that you can use a strain from the literature somewhere or a taxonomic relatedness. No, it's really the progenitor. So a strain from the same lineage, so that we can assess the steps which have occurred between that strain and the other one.
Yeah. Okay. Thank you. That answers the question. Okay.
Okay. Good.
Okay.
Sorry. Let me try a couple of hands then. We have Natalie Hennin. Do you want to ask on the same topic, or should we wait? I have a hand raised. No.
Can you hear me?
Yes. Yes.
Hallelujah. Sorry. Now, my question was close to the first question, but a little bit different. Because it relates to two enzymes produced by a parent strain, but which has undergone different mutagenesis steps, non-GM, of course. And would it be possible to do only one subchronic study and compare non-sequences for biosynthesis of secondary metabolites and or mycotoxins? Then if we do that, could we do only one subchronic toxicity study?
That's back to you, Lever, I think.
I'm not a toxicologist. I'm a microbiologist, but when I consider what can happen by conventional mutagenesis, like I explained already, I would suppose that we can only rely safety on a full set of toxicity studies. But I can ask the toxicologist colleagues what they think about that. Claudia? Oh, yes, Andrew.
This is the view. I mean, any toxicological studies are really strain related. Anytime that if you make, as Liv has clearly stated in the previous response, if you make something in which you have a very well recognized parental, and then you can follow the modifications that has been introduced, for instance, by cloning a single gene in a defined locus, indicates a situation of very related. But if you enter with mutagenesis, which is a random change in the genome level, you can expect some activation of new or deactivation of some important pathway. So it is, in this case, a new tox data should be provided.
Yes, because the toxicological study try to address the secondary metabolite, the complex compound present in the final product. So I think we need a specific toxicological study. I agree with your answer, Liv.
Thank you, Claudia.
Okay. So we have another question from Bas Verhagen.
Yes.
Same topic, I think?
Yes, it's a similar topic at least.
Yeah.
It's the topic of the first presentation still.
Thank you.
My question is regarding the FASTA files. As you are all well aware, and I can see from the smiles, that the FASTA files are very sensitive to industry, and having them somewhere is for industry a high risk. And that combined with the new transparency regulation, which actually makes it less certain that they can be kept confidential. We were wondering if the applicant provides all the information to show exactly how the whole genome sequencing and all the assessments were performed, if it could not be possible just to not make the FASTA file submission a must, but more keep that on a case by case, if there are still unclarities left at the end.
Yeah, I was smiling because I was absolutely waiting for this this question, and actually was raised by several commenters in the open consultation. But the answer in this case is pretty simple. Let me say that I cannot enter in the question of confidentiality. I will leave to the EFSA officer addressing this part probably tomorrow. I think is also one of the topics that should be discussed. But in terms of risk assessment, the availability of FASTA file, it is important. I have to say that now it's even more, the requirement is higher because not only the FASTA file, but also the raw data deriving from sequencing are required. But at this time it's not our responsibility because there is this overarching statement that applies to all microorganisms intentionally introduced in the food system, such as food enzyme producer, feed additives, plant protection product, and so on. So it covers all the microbial application under the remit of EFSA. And these statements require the submission of all the set of data related to the whole genome sequence.
So we're not really hiding behind other people. It's true that we are simply being consistent with other elements in EFSA. But I agree with you that if EFSA demands commercially sensitive information, then the onus is on EFSA to guarantee to protect that information.
Yeah.
And I mean, EFSA is a responsible organization. And I'm quite sure it will make great efforts to do just that, to protect sensitive information. But well, I guess it's wait and see, isn't it? So, E, do we have anything else on the microbial area, or can we move on to genotoxicity?
We have actually one question about the substitute toxicity. So
okay, let's take that.
Okay, Bas, please.
Okay.
Hello. I think that question was pretty much answered because I was asking if mutagenesis could be an option, but I think it was quite clear that as soon as you use mutagenesis, this will not be accepted to use as a separate tox study.
Yeah. Okay, so if there's nothing else on the microbial elements, can we turn to genotoxicity? E.
Okay, so there is one comment from Joy Hardinger, and can you please speak up?
Thank you. It wasn't on the genotoxicity, it was on the toxicity dossier.
That's fine.
My question was regarding an applicant putting in a dossier, say for a plant enzyme, where they have put forward a case that there is an evidence of consumption, etc. So they have not included toxicology. But when the EFSA panel reviews it, they decide toxicology is needed. I'm asking whether the panel would then come back to the applicant, ask and give them time to produce the toxicology before the opinion is published, because that would make quite a large difference to the applicant. Thank you.
I mean, first of all, let me say that if EFSA raises a question with an applicant, there is no possibility of our producing an opinion or submitting an opinion in the interim period. We would always wait for the answer to any question if we've raised one with an applicant. That's in principle, but I'm not quite sure the circumstances under which we would ask for toxicology data for a plant enzyme where we have evidence of consumption or equivalent consumption. I wonder, I mean, E can...
Yeah, of course, Andy, there could be an option if the enzyme is not derived from an edible part of a plant, or if the consumption is significantly lower from the traditional source as compared to the use of the enzyme. This would be two options where we could, in theory, ask for toxicological data.
Yeah, I think I also would like to add that from the chat, I can read that the applicants believe that it's not necessary to carry out toxicity tests, no? So it's exempted. And I think in that case, and EFSA will stick to the principle that the applicant has a right to be heard, meaning that, and during the evaluation, and like the case Holger has given the example, in case EFSA believes that a toxicity study should be carried out, and EFSA will ask this question. And then I think Andrew's answer comes in, and we will wait for your answer and before finalizing the opinion.
Thank you very much. That's very helpful.
Okay. Andrew, that's all the questions, all the comments. There is nothing more.
No, you surprised me. Nothing at all on allogenicity. Nothing at all on exposure estimates.
Not at all.
Okay, this could be your last opportunity for a year to ask questions of members of the working group. No?
There is one hand.
We have one hand. Yes, got you. Okay, that's Hagen.
Yes, thank you. I wouldn't let the opportunity go to ask one more question.
Oh, please.
It was stated that we, as applicant, need to look at the whole genome sequencing and compare it to at least 2 up-to-date databases. The question is what can the applicant do if there are no 2 up-to-date databases? And especially for the small and medium enterprises, some of the databases available are already really expensive. So finding 2 that are properly up-to-date and not too expensive, that might be an issue. So I was wondering if there's any way that can be reduced to 1 database.
The choice of the first question, these databases are not expensive in the sense you can either do online the analysis, or if you have any confidentiality issue, you can download by all the set of gene and make on your own computer, because it's something like in the widest contains 6,000 genes, so it doesn't not to take too much time, so are totally free. And also the program for making the alignment of sequence, there are free tools. And having 2, I think, is important, because some of them, they use different methodologies in comparing sequencing. So I think it's a sort of safe, but it is not an issue of cost, really. The databases are totally free. You can download on a variety of websites, institutional website like NCBI, MBL, or specific like CARD or ResFinder, which is maintained by universities. So there is no cost issue on this point. and two, just because the system used to detect the genes is different, so it's better to have two. And generally, this is also in favor of the applicant, because if only one can detect the gene, you can have a look at what kind of homology has been found, or what is the type of gene, so it's better to. It is not against the applicant, but let me say it is in favor of a better knowledge of the strain under assessment.
Does that answer your question, Lars, or would you like to come back?
Yes, just for my clarity, these databases are examples mentioned in the previous statement.
Sorry, the sound was disappear for a while. What was the question?
Examples of these databases, I was just wondering, are they mentioned in the previous statement?
Yeah, the problem that one of the previous statement was no more updated. They decided to join with another. So if I remember well, is arg anot is no more updated. So, but there are, I think we have seen in these last 2 years a number of applications, and most of them they use updated databases. CARD and ResFinder is just two of them.
But can I make the point that we will not specify in guidance which databases can be used, because as Sandro has just said, circumstances change, and it's quite likely that a database we recommend suddenly ceases to be maintained or just disappears. So by and large, we will not give examples. We expect applicants to use the most updated information. Okay, so there's another hand raised, Melina Galano.
Yes, good afternoon, Melina Galano from DSM. Thanks a lot for all the presentations. I actually have a question on a topic that was not addressed today, so okay. I will just ask the question and see if it's possible to answer. Yeah, that's about the 90-day toxicity studies. And in the updated guidelines, it says that the highest dose selected should be at least 1,000 milligram TOS per kilogram body weight per day. And yeah, I was wondering if someone could explain a bit the reasoning of this highest dose. And maybe my question behind it is that in some cases we don't feel that we need to go that high. In some cases, we actually need to go higher to cover all our applications. So, yeah, is there a way that we can justify a lower dose, for instance? What I have in mind is also older toxicity studies for which we didn't necessarily dose that high. And I hope that they will still be accepted even if the highest dose was not 1,000 milligram dose per kilogram body weight.
I think we've already agreed that this guidance will not come into force for some time, and therefore we have to accept data which was produced under other circumstances and other guidances, and that's a general rule. So I think you can be reassured that your 90-day studies will not be simply rejected because you haven't used a gram top dose. I will make two comments on this and then pass it over to people who really understand toxicity assessments. One of which, the 1,000 grams comes essentially from the OECD limit dose. And it says that it argues that where you cannot demonstrate any evidence of toxicity, because ideally in a 90-day study your top dose should show some element of toxicity. Where this cannot be demonstrated, then the OECD recommends that you study just one limit dose of 1,000 milligrams, one gram per kilogram body weight. That's fine, except that this was a system that was designed for the assessment of single chemical items and not for mixtures. And so it may be that we have problems with 90-day studies which have a relatively low top dose when we know that we're already dealing with a mixture, and what may be potentially toxic may be so dilute within that limit that in the lower dose, that we would never ever see a response. So if we have at least 1 gram per kilogram body weight, at least we have a reasonable chance of detecting a potentially toxic material. Now that would be my understanding, but perhaps I pass it over to Claudia or to Henk,
No,
who can give a much better answer than I can.
I perfectly agree with your answer, because there is the same problem also for genotoxicity test, because we have a mixture, so we need to reach enough concentration also for the minor component of the mixture. It's the same observation I presented for the in vivo studies for genotoxicity test, because in vivo we have this problem. In vitro we can try to understand something more, but in vivo we need to give to the animal the maximum concentration is possible in order to have the possibility to detect any toxic effect as possible.
Yeah, I have little to add. This is exactly the way you say it, Claudia, and also Andy.
Okay.
Okay, thank you.
So, do we have any others? E?
Well, I don't see hands raised, and
Neither do I.
maybe you can proceed to wrap it up.
Okay. Well, that could be very quick. I could just say good night. No, I'm not going to spend a lot of time. I really would like to thank you all for listening patiently to us, to some of the questions you've raised, and actually for just being willing to participate in this event. I started out by saying we had a number of principles or objectives we hoped that our guidance would meet. I'm still not sure whether you actually think we've achieved those objectives or not, but I hope you do. I also hope that you're going to be with us tomorrow when you will learn, as I said, the first item on the agenda is one for which we have little experience and really would need feedback from you, and that's the question of modifications to existing authorizations, the guidance part. And you will learn a great deal about the new administrative procedures for new applications for dossiers. And the other thing I would like to say is that as a working group, we have seen a dramatic change in the dossiers that have been submitted. The ones that have been submitted in 2020, 2021 are much, much more closely aligned with the sort of requirements we have in the revised dossier or in the various statements we've introduced in the past. And I thank you for that, because it really does make a big difference. The trouble with the older dossiers, the first 300 submissions is, of course, they were written at a time when all of us were starting out on this journey, and it was inevitable that there would be a fair amount of dialogue in each case to achieve a suitable end. I'm pleased to say that with the dossiers we have now, I think the need for that dialogue has been substantially reduced. And I really must congratulate the applicants on the quality of the dossiers that they are now producing. So may it continue in this route, and perhaps we have some chance of working through the several hundred dossiers that are currently facing us. Having said that, I hope you will all be with us tomorrow afternoon. And if there are no other comments, I wish you a good evening, good night, and thank you for attending.
Thank you very much.
Bye-bye. Have a nice evening.
Thank you very much.
Thank you.
Enjoy your
Bye-bye.
evening.
Goodbye.
Bye.
Thank you, everyone.
Bye.
Bye.
Bye bye.
Okay, bye-bye.